let's, I think, kick off now. So without further ado, let me introduce my colleague, Andrea Luppi. Andrea Luppi is a PhD student in clinical neurosciences, working in the Division of Anesthesia in the Alan Turing Institute. He comes from a background in philosophy and cognitive science, and his work aims to characterize how the capacity for cognition and consciousness arises from the complex interactions between brain systems viewed as a network. He combines tools from information theory and network science to study brain activity across altered states of consciousness, such as anesthesia, disorders of consciousness, and psychedelics. And his talk today is called Mapping the Landscape of Consciousness and the Dynamics of the Human Brain. Is that right, Andrea? Excellent. So I think you should be able to share your screen.
Not yet.
Not yet. Okay, momentarily. Ali, you might need to make Andrea host.
Done. That should work. Now.
Over to you, Andrea.
All right. Let's see. Can you see my screen now?
Perfect.
It should go into presenter mode in just a second. Right. So yeah, thank you so much to the organizers for having me. And as Henry was mentioning, the topic of my work is really about consciousness. And the reason why I think it might be of interest to people in the audience today is because often when we talk about consciousness, we are talking about something that we cannot directly observe. And we typically rely on behavioral reports to know whether someone is conscious or not by asking them to perform a task or to simply say, squeeze a clinician's hand. But often, as it happens, there might be cases where that is not possible. And we'll deal with that in this kind of research. So let's get started. And as I mentioned, the interest in consciousness, according to some people, might be almost paradoxical as a topic of scientific study, because as William James famously described it, it is akin to the flights and perchings of a bird. And what I'm trying to argue in this presentation is that we can actually make inroads into the study of consciousness by understanding how at least some brains work. And the hope, of course, is that this might generalize perhaps beyond humans, although I should be clear that I will only be talking about human brains so far. And so really what we want is to avoid this particular situation and rather turn the understanding of consciousness from magic into hopefully science. And the approach that we take is conceptually relatively simple, because we start from a philosophical question, arguably, which is how does mind arise from matter? And we turn it into a scientific question by asking how brain activity and connectivity support consciousness. And we do it by simply scanning people under two different conditions, one in which we think and we have good reason to believe that they are conscious, and one in which we think that they are in fact unconscious. And we try to see what differences are there in their brain activity or in their patterns of brain dynamics, which is what I'll be arguing about today. And really, this simple approach is made more complicated by the fact that people can lose consciousness in a variety of different ways. We're all familiar with loss of consciousness as a result from a good night's sleep. But sometimes consciousness can be suppressed by means of an anesthetic, which is required for surgery. And of course, there are different kinds of anesthetics which operate through different kinds of molecular mechanisms. And in more unfortunate circumstances, someone might suffer some kind of traumatic or anoxic brain injury and become chronically unconscious as a result and develop what is known as a disorder of consciousness. And all of these are different paths to the same state, which is unconsciousness. And what we're trying to characterize in the group where I work and in my own PhD work is what is similar across these different ways of losing consciousness. And in particular, the focus has really been on induced unconsciousness, whether that be induced by anesthesia, which is pharmacological and reversible, or whether that is induced by brain injury, which is not pharmacological. It involves structural changes in the way that the brain operates, and it is often chronic as a condition. And one interesting aspect of brain activity, of course, is that brain structure remains relatively constant across short time spans. Over the span of, say, a few hours, the brain structure remains the same. And nevertheless, your brain patterns and my brain patterns and my mental states and yours, they change much more rapidly. They change over the course of a few seconds, sometimes even shorter than that. So how is that possible? How can a static structure generate dynamic mental states? And of course, the question is something that we think we can address in terms of turning mental states into brain states, because we cannot really observe mental states in others, but we can observe their brain states using brain scanning, and we can try to relate those to the underlying structure of the human brain. And the way we think about this is really in terms of relating structure and function. And in turn, you can think of them as the roads between different parts of a city, that being the structure of your brain. And the function can then be thought about in terms of traffic that occurs over those different roads. And that is something that unfortunately we're all fairly familiar with. But what we also know is that traffic is not constant across time. So even if the roads are the same, sometimes the traffic on them will be very high. And so a larger road will have more traffic on it. But at other times, for instance, during the weekend, that might not be the case. So even if a road is very large, there might only be little traffic over. And this is an important point because we think that this is very similar to how the brain actually operates. Structure and function can be mapped onto each other, but that mapping need not be constant. And in fact, its changes may reveal something about what is going on in someone's mind. And this is a formalization of how we approach this problem. So effectively, you could see us as mapping cities onto different brain regions. And then thanks to diffusion MRI, which is a non-invasive methodology, we can map the structural connectivity between different brain regions in terms of the presence of white matter pathways between them. And likewise, we can use functional MRI to obtain signals of brain activity over time coming from each region. And that lets us have both the roads and the traffic. And so now we'll see one particular way that we can relate the roads and the traffic and quantify their relationship. And I think the best way to describe it is with this short video, which hopefully you'll be able to hear and see as well. Now, hopefully you were able to hear the various vibrations that were happening as well. And this is a formal description of what was happening here. Basically, as specific frequencies were being transmitted over the metal plate, the sound that was present over that metal plate was reconfiguring itself. And the reconfigurations were far from random, but they were also related to the frequency, so that a higher frequency produce progressively more complex patterns. Now, at this point, you might be wondering, we were talking about brains, now we're talking about sand and metal plates. What do they have to do with each other? But actually, it turns out that we have very good mathematical descriptions of how these patterns of sand form themselves according to specific frequencies. And this is what is known as harmonics. And there are mathematical definitions of these that we can use. But this is not just something that pertains to abstract concepts and very experimentally contrived scenarios like sand and plates. This is actually exactly how we make music. This is why it is called harmonics, because the way that we make music is related to generating different kinds of frequencies over, say, the body of a guitar, which correspond to different patterns of vibration and different patterns of activity, if you will, of the body of this guitar. And in fact, this generalizes even further because it has been shown that this is also how animals get different kinds of coat patterns over their fur. And so if we take this well-formalized mathematical framework we can then generalize it to the human brain. We can replace the metal plate with the structural connectivity of the brain, so the roads in the brain, if you will, and we can replace the patterns of how sound organizes over that metal plate with the patterns of how brain activity organizes over the structure of the human brain. And this is what is known as Connectome Harmonics, connectome being the word for the structural connectivity of the human brain. And as you can see here, there is a remarkable analogy between the harmonics that we observe over sand in a metal plate and the harmonics that we actually observe in the brain. And to give you a slightly more formalized version of that, this is the actual procedure that we use. So very briefly, we combine information about anatomy and connectivity in the brain, and then we use exactly the same mathematical framework, which is called Laplacian eigendecomposition, for those of you who care about the math. And we obtain progressively more fine-grained patterns of brain activity that develop over the same structural connectome, the connectome of the human brain. And it is important to note that these are progressively more fine-grained because this will be the way that we interpret these patterns. And very briefly, we can use this mathematical framework to decompose brain activity so that at each point in time, we re-represent the brain activity that we observe into a combination of these harmonics. Now, for those of you who are native speakers of math, this is effectively a generalization of the Fourier transform to the connectome structure. Now, I myself have a background in philosophy. I am not a native speaker of math. So I like to think of this in terms of smoothies, which is basically to say, imagine that you have two different smoothies and you're trying to figure out which of those finite set of ingredients went into each smoothie. You could roughly imagine that the one that has a higher redness is probably involving more watermelon and more strawberry. And the orange one instead involves more orange and more mango. And this is just the same exact principle that we use. We see a particular pattern of brain activity, and we try to understand with mathematical tools how much contribution each of these harmonics made to this particular activity over time. So the harmonics become the alphabet, if you will, that we use to construct these brain states. And as you might remember, we are using brain states as a proxy for the corresponding mental states. And this framework, which was actually not developed by myself, but by Selen Atasoy and her colleagues, this framework can be used, finally, to study consciousness. And in particular here, what we are showing is that there is a difference between the kinds of patterns of harmonics that we observe in the brains of participants who are under the effects of a placebo or under the effects of LSD, which is a serotonergic psychedelic. And just to show in a different way, what we are seeing is that the contribution from low frequency harmonics, which are the very coarse-grained patterns, becomes reduced when you're under the effects of LSD. And instead, your brain activity is increasingly comprised of highly complex, fine-grained patterns of harmonics. And the same group of researchers was also able to show that this signature is not just a signature of LSD, but it is also a signature, the very same signature of the serotonergic psychedelic psilocybin, which is the compound found in magic mushrooms. So this is not specific to a given drug, but it is at the very least specific to a given class of drugs, the serotonergic psychedelics. And this is where my own work comes in, because we were able to show that this signature generalizes even further to a sub-anesthetic dose of ketamine. Now, the reason this is interesting is that ketamine, unlike the other two, is not a serotonergic psychedelic. It operates very differently in terms of its molecular mechanisms of action. And importantly, it can have different effects depending on its dose. A very high dose of ketamine is actually a dissociative anesthetic. But a low dose of ketamine, which is what we use here, instead acts as what is known as an atypical psychedelic, meaning that it is not a classic serotonergic psychedelic, but subjectively, it is similar to the effects of a psychedelic. And this is what's important here, because from this slide, we know, and from this set of results, we know that what these harmonics are tracking is not just a particular molecular mechanism of intervening on your brain, but it is actually more closely related to the subjective mental state that these different classes of drugs can elicit in a participant. And of course, we've controlled for the silly confounds such as motion, so we know that that's not just that. And interestingly, what we were able to show is that the very opposite signature is observed when people become unconscious as a result of anesthesia with the GABAergic anesthetic Propofol. Very opposite signature, meaning that the more complex patterns of harmonics become suppressed, and instead brain activity is increasingly explained by structurally coupled, coarse-grained patterns of harmonics. So the less complex ones become increasingly prominent. And what I think is especially interesting here, though, is that we were able to generalize the same pattern observed from anesthesia to disorders of consciousness. Now, why this is interesting is first, because we are able to say that once again, this signature is not a signature of a particular anesthetic. It's not even a signature of anesthesia in general, but rather it is a signature of unconsciousness, because we can observe it even in people whose unconsciousness has been the result of a very different perturbation of their brain, namely a structural, physical perturbation. But the other twist that is present here is that these are not patients compared against controls. These are two different groups of patients that we are comparing against each other. So this signature in particular is obtained by comparing patients who are unresponsive and patients who are unresponsive behaviorally, but nevertheless are able to perform the so-called tennis task in the scanner. So let me describe that a little bit more in detail. We know that these patients are all diagnosed as having a disorder of consciousness because they cannot perform motor responses when they are asked by the clinician. The clinician might ask them, squeeze my hand or follow me with your gaze, and the patients will not be able to do so, and they will be diagnosed as having a disorder of consciousness. However, you can imagine that inability to perform the required task being not because the patient is unconscious, but perhaps because the patient is unable to move because they have a damage to their motor regions, for instance. And in 2006, Adrian Owen and colleagues were able to show that if you ask some of these patients not to play tennis, not to move around, but to just imagine playing tennis while lying in a scanner, the corresponding brain regions that would activate in my brain or in your brain when we imagine playing tennis would also activate in these patients' brains. And so some patients, and this is it not just specific to tennis, but rather also other tasks where we know very well what regions we would expect to see activated, such as navigating around your own home. When that happens, we can tell that some patients, despite being overtly unable to respond to commands, in fact, they are covertly conscious because their correct set of brain regions activate when they're asked to perform these tasks. And so this is what we are comparing here. We are comparing all two sets of DOC patients. One set could not perform this task, so we have no evidence that they are conscious. And the other set of patients, 8 of them out of 20, were in fact covertly conscious. And this signature is differentiating between these two sets of patients. So it's really quite specific to consciousness rather than just being due to the presence of rather severe brain lesions, as you might expect. And we were also able to show that this signature can generalize. So the similarity of a given healthy individual's brain activity to the pattern obtained from the patients will predict how much anesthetic is in their blood when they become unconscious, or vice versa, when they regain consciousness. So this is really quite generalizable across different data sets. And in fact, we showed that the same happens when you take the similarity of someone's brain activity to the pattern obtained from ketamine, again, psychedelic levels of ketamine, and that will predict how that person's brain activity is relating to the LSD experience. So, and will predict how intense they are feeling the LSD experience to be. So this is generalizing from a brain state to the corresponding mental state that the person is experiencing at least at the very coarse level of how intense the psychedelic experience feels. So just to wrap up and leave some time for questions, what we've been showing here is that there are clear signatures of the psychedelic experience that are not specific to how it is induced, and that we can obtain them by looking at brain activity over time and how it relates to the underlying connectivity of the brain. And we've also shown that the very opposite signatures is obtained when looking at unconsciousness. And once again, doesn't matter how that unconsciousness comes to be. It can be induced by anesthesia, it can be induced by a disorder of consciousness, but so long as it is unconsciousness, the very same brain signature that we observed from psychedelics is observed, but reversed. So instead of being more complex, the pattern is less complex. And we can actually formalize this in terms of looking at the entropy of this pattern of harmonics, which is basically just a measure of their diversity. And what we see is a very nice one-dimensional continuum, whereby at one end, we have the comparison of patients with a disorder of consciousness with controls, or with covertly conscious patients. And at the other end, we have the effects of different psychedelics. And as you will note, there is also here a particular data point pertaining to different levels, comparing moderate propofol sedation with mild propofol sedation. Now, why this is interesting is that mild propofol sedation means that participants already have the drug in their system, but that is not sufficient to make them unresponsive. So this is not just tracking the presence of a drug in your system, even an inhibitory, an anesthetic drug, but rather the signature only appears when you lose consciousness. And of course, we also show that the signature is reversed when consciousness is recovered in these same individuals. So it is not just tracking what goes away, but it's also tracking the recovery of consciousness when that happens. And so the hope of this talk really was to show that we might be able, simply by looking at brains and their activity over time and how it relates to their structure, their connectome, to understand something about what kind of experience is going on in those brains and what kind of consciousness or lack thereof is present there. So if you will, we can use these harmonics as a tool for mapping the landscape of possible states of consciousness. And of course, it is a different question to what extent this can generalize to other species where we don't have the ability to access direct reports. But I think this can help us make inroads into what it is that makes a brain conscious or unconscious or in an altered state of consciousness, and get us closer to relating consciousness, cognition, and the brain. And of course, I have to thank very many people, including my funders, but also very many collaborators who have provided insight and data and expertise into making all of this work possible, including my own membership of CFI and the Alan Turing Institute, where I am at the moment. And really all of you for your attention, and the organizers for giving me the really valuable opportunity to present this work to you. And you can find all of these results into a preprint that is open access, and it's linked here at the bottom. And I would be delighted to take any questions that you have. Thank you.